The overall goal of this nanostructured chip-based platform is to analyze membrane protein function on the single molecular level in a highly parallel manner. Through its ability to resolve single proteins, new insights into membrane transport and flux are achieved.
This method can help answer key questions in the membrane protein field, such as the conductivity or transport behavior of single channels or transporters, especially such with non-electrogenic cargo. The main advantage of this technique is its high parallelism, derived from nanostructured chip design, featuring hundreds of thousands of nanopores and the fluorescent readout of transport events.
The implications of this technique extend towards screening-based assays of effector libraries for membrane proteins, because the chip-based platform enables highly parallel and automated transport recordings. Generally, individuals new to this method will quickly learn how to handle the presented technique. The focus was always to create a user-friendly assay so that everyone interested can adopt it.
This membrane transport assay utilizes a silicon-on insulator wafer, engineered using reactive ion etching. About 1,000 assay chips are fabricated from each wafer. Each chip contains 250,000 microcavities. These cavities get sealed by lipid bilayers containing the transport proteins of interest. The membrane constitutes the sensor interface. Initially, fluorescent materials are contained to one side of the lipid bilayer, corresponding to the interior of the light.